Hello everyone. Today we will discuss about the development of pharyngeal apparatus, or also called as branchial apparatus. So why this pharyngeal apparatus development is important? Because many babies will be born with congenital anomalies like Treacher-Collins syndrome, which is also known as the first arch syndrome. There is a Robin sequence, golden heart syndrome, and there are also many congenital cases like the unilateral cleft lip with cleft palate, bilateral cleft lip with cleft palate. And to understand the facial anomalies like you see here, again cleft lip with cleft palate and partial cleft palate, and there is a oblique facial cleft in the face, and there is a median cleft lip. So before going into the details of pharyngeal apparatus, let us try to understand the folding of embryo. As you all know, the folding of embryo will start around the beginning of the fourth, that is end of the third week, beginning of the fourth week, where a trilaminar embryonic disc is converted into a cylindrical embryo. That is the purpose. So during the third week, there will be formation of the three germ layers called as gastrulation. The three germ layers are ectoderm, mesoderm and endoderm. From above downwards, that you see, there will be folding of this disc into a cylindrical embryo. You can see the head and tail folds here, head folding and tail folding, and there is also simultaneous lateral folding. So this folding processes convert the flat disc-shaped embryo into a cylindrical embryo with the cavities. You can see here there is a amniotic sac above and yolk sac below, and due to head fold and tail fold, they will result in following changes. Amniotic cavity will enlarge, yolk sac cavity becomes smaller, and it becomes intraembryonic yolk sac, and there is a yolk stalk and extraembryonic yolk sac. Allantois connecting stalk shift caudally. Septum transversum comes anterior to cardiogenic plate. It comes anterior to cardiogenic plate. Let us try to understand the folding with the animation here by the medicovisuals.com. I suggest you to visit this site and try to understand embryology in a better way. You can see there are three germ layers. Ectoderm is denoted in blue color, mesoderm in green color, and endoderm in yellow color. You can see the two areas there. That is the oropharyngeal membrane and cloacal membrane, where the cells of ectoderm and endoderm are adherent tightly without any intervening mesoderm. If you just see here, that is the towards the head end called as oropharyngeal membrane, and towards the tail end we can see cloacal membrane. If you zoom here, you can see the cells of blue colored ectoderm are adhered to the cells of yellow colored endodermal layer here. So there is no intervening mesoderm. These are the only two sites in the embryonic disc where there is no mesoderm intervening. So this oropharyngeal membrane and cloacal membrane will decide the head end and tail end, and they provide the access for the folding. First the head fold will start. You can see above the ectoderm you can see amniotic cavity, and below the endoderm you will see yolk sac cavity. So the folding will start. As we discussed earlier, what is the purpose of folding? To convert a disc-shaped embryo into cylindrical embryo. So to give the body a shape, as you know, human body is cylindrical with outer ectoderm, intervening mesoderm and inner endoderm. So this is the beginning. Here you are seeing the two lateral folds. You can see these are two lateral folds from the side folding, from the sideways the folding is happening here. So because of this, these germ layers will also undergo folding and they form tubes. You can see endoderm is folding there. So this is the lateral folding, and there will be also simultaneous head and tail fold. If you see the sequence there, first there will be head fold, then tail fold. So like this. So entire embryonic disc is converted into cylinder which is covered outside by the ectoderm, that you can notice there. So because of this, there will be head fold and tail fold. So the innermost endoderm will convert into a primitive gut tube. That part of the gut tube in the head fold is called as foregut, in the tail fold is called as hindgut, and in the middle part it is called as midgut. So this is how the folding happens, and this is how the cylindrical embryo is formed here. Now if you take out the outer ectoderm, you will see the middle mesoderm. You can see the mesoderm here, forming the intervening layer between ectoderm and endoderm. And once you take out the endoderm, you will see a tube like endoderm. Now this tube is called as what? Primitive gut tube, which is forming the entire digestive system, that is gastrointestinal tract. So this is how it is there. And we have learnt earlier that there are two places where there is no intervening mesoderm, that is oropharyngeal membrane towards the head end, cloacal membrane towards the tail end. This is just a coronal section. Now we will take a sagittal section of this cylindrical embryo. After removing the ectoderm and mesoderm, you can see endoderm here, that is forming a tube, primitive gut tube, and it is in contact with the ectoderm at oropharyngeal membrane in the head end and a cloacal membrane in the tail end. They form respective oral orifice and anal orifice in the developing embryo that will study in the development of GIT. So this is how the disc-shaped embryonic structure is converted into cylindrical embryo. You can notice. And below what you see, a reduced yolk sac. Most of the yolk sac becomes intraembryonic and forms primitive gut tube and connected to the extra embryonic yolk sac via a yolk stalk, that we say. And above what you see is the enlarged amniotic sac. So coming to the development of this pharyngeal apparatus. So these mesenchymal cells are involved in the formation of head and neck. What are mesenchymal cells? They are the loose connective tissue cells derived from the paraxial and lateral pit mesoderm and neural crest cells, and they form the different components of the head and neck here. You can notice there, neural crest cells form the most of the facial skeleton, whereas the paraxial mesoderm forms the most of the neurocranium along with the muscles, and lateral pit mesoderm forms the laryngeal cartilages. So entire head and neck is derived from these mesenchymal cells. Okay, so the mesenchymal cells from three sources: lateral pit mesoderm, paraxial mesoderm, and neural crest cells, along with ectodermal thickenings called as ectodermal placouds. So they form different parts of the head and neck. Not only these skeleton and muscle, they also form these dentine, tendon, dermis, ligaments, meningeal layers, sensory neurons, and glandular connective tissue. So these all are formed by the mesenchymal cells coming from these 3 to 4 structures. What are those? Again we will repeat: paraxial mesoderm, lateral pit mesoderm, neural crest cells, and the ectodermal placouds, and they develop into pharyngeal arches. The pharyngeal apparatus is also known as branchial arches because it is similar to gill slits in a fish. So coming to the development of neural crest cells, as we know, the upper ectodermal layer will form a slippery shaped medullary plate or neural plate. In the midline it will form a groove. That groove deepens to form the neural tube, and these lips of the neural groove, when they join, there will be group of cells between the overlying ectoderm and neural tube. They are called as neural crest cells, and neural crest is also referred as the fourth layer of the embryo along with the three other germ layers. You can see there. There's a overlying ectoderm, then there's a neural tube. So neural tube is derived from the neuroectoderm, which is derived from the medullary plate or neural plate that you see in the center of the slippery ectodermal layer when the embryo is in a disc form. So you can see there. So these neural crest cells have a significant contribution in the development, and you can see the neural tube will develop into three brain vesicles: forebrain, midbrain and hindbrain. And above the neural tube and below the ectodermal layer, you will see neural crest cells which migrate from dorsal to ventral into the pharyngeal arches, and they help in the formation of the different components of pharyngeal apparatus. Mainly we discussed facial skeleton. Facial processes are derived from the neural crest cells, and when they migrate from dorsal to ventral, they form these thickenings called as pharyngeal thickenings, which in future form pharyngeal arch. You can notice there, if you take a section there, so after the folding in the embryo, you will see two tubes there. So overlying neural tube, which is involved in the development of the CNS, you can see there in this view, you can notice. So this is the cylindrical embryo. Outside ectoderm is there. Purple color area is neural crest cells, that is neural tube, and yellow colored primitive gut tube. And in between them, you can see there is a mesoderm, and these neural crest cells migrate dorso-ventrally from posterior to anterior, or in the embryonic terms from dorsum to ventral around the foregut, because we are seeing the cranial part of the gut tube here, foregut, and in between them the mesoderm, the intraembryonic mesoderm, will differentiate into three parts. Paraxial is in pink color, then in the white color you can see intermediate mesoderm, then in the brown color lateral plate mesoderm. The lateral plate mesoderm will fuse in the midline because of the lateral foldings. So here, as we discussed, the pharyngeal apparatus has contribution from 3 to 4 sources. So mesenchymal cells coming from neural crest cells, paraxial mesoderm, it forms somates and somatomeres, and lateral plate mesoderm, which will form the most of the soft tissues of the head and neck. So mesenchymal cells. What are mesenchymal cells? These are loose connective tissue cells. So from here you can see, so all these mesenchymal cells along with neural crest cells, they migrate or they move from dorsum to ventral between the oropharyngeal membrane and this foregut that will form future pharynx. So these are surrounding the pharynx, that's why they are called as pharyngeal arches or pharyngeal apparatus, not branchial, because we don't have gill slits like in fishes, but it is also called as that, just a reference, evolutionary reference. So here you can see, so all these mesenchymal cells and neural crest cells, they together have a role in the formation of pharyngeal arches, which is covered outside by the ectoderm, which is marked in the blue color, and inside by the endoderm, that is marked by the yellow color. So they are surrounding these thickenings are surrounding what? The primitive pharynx coming from the foregut, because foregut is the part of the primitive gut in the head fold. Okay, that's why they are called as pharyngeal arches or apparatus. Pharyngeal arch is mesodermal, you can see there. Okay, so inner layer is endoderm, outer layer is ectoderm. In between them, what you see? Neural crest cells, mesenchymal cells coming from paraxial mesoderm, and lateral plate mesoderm. These are forming the arches, and these are giving rise to different structures of head and neck that you can understand here. So this is how it happens, and you can see finally they form these thickenings between the developing brain and face and around the pharynx, ventral and lateral to pharynx. or you can say simply anterolateral to pharynx. These thickenings are produced on both the sides, and this is how the pharyngeal apparatus has the what you can say resources in these mesodermal and neural cells. So these surface elevation appear in the lateral wall of the primitive pharynx, caudal to stomodium, that is developing oral cavity. So it consists of the pharyngeal arch and depressions on the outside called as clefts and inside called as the pouches. So initially they are around the primitive pharynx, but gradually they extend ventrally and form a horseshoe shaped cylindrical bars. So initially there are 6 arches, the 5th one will disappear. So they form these 5 arches, 1, 2, 3, 4th and 6th. The 5th arch disappears. So this is how you see the horseshoe shaped arches are formed here. So 1, 2, 3, 4, 6. And what are the different components here? So pharyngeal arches are 5 in number, and whereas pharyngeal clefts, which are formed by the ectodermal invagination, they are called as the pharyngeal clefts. They are 4 in number. Pharyngeal pouches are endodermal evaginations, outpouchings of endoderm. They are also 4 in number. And where this ectoderm and endoderm meet, they form membranes. They are called as pharyngeal membranes. So pharyngeal apparatus contains 5 arches, 4 cleft, 4 pouch and 4 membranes. How? Let us try to understand with this animation here. We can see these are the surface elevations formed around the developing face there. There is a frontonasal process, and there is and these are pharyngeal arches, you can see covered outside by the ectoderm. That's the first arch, having 2 processes of ventral maxillary and dorsal mandibular processes. Like this, these surface thickening will appear, that is the oropharyngeal membrane forming the future oral cavity. Hope you are following the animation here. You can see that is the first arch with the maxillary and mandibular process. Second arch is there, third arch, fourth arch, and there is 6th arch. These are the elevations on the side of the developing primitive pharynx, that is the neck there. As you see pharynx in the neck, you can imagine them in the neck region on either side, right and left. And they fuse to form what? The horseshoe shaped arches, covered outside by what? Ectoderm, inside by endoderm. That endoderm is what? It's a part of foregut, that is forming the primitive pharynx there. If you take a section there, what are the layers forming these different components? You can see outside what we have seen is the ectoderm, then we have the mesoderm. You can see the ectodermal clefts there, 4 clefts. What are clefts? These are ectodermal invaginations. So these are clefts there. You can see ectodermal cleft, 1, 2, 3, 4. And if you take a section like this of the cylindrical embryo, take out the superficial, the ectoderm, then you have mesoderm, then you have a developing neural tube that forms the brain and spinal cord, and beneath that you will see developing foregut. So in the previous section we have seen the clefts, you can see now we have removed the neural tube here, just that is one arch, you can see there, that is one arch which was shown in the square box. So outside you have ectoderm, inside you have endoderm, in between you have mesoderm. You can see there are ectodermal clefts and endodermal pouches. Clefts are invaginations of ectoderm, whereas pouches are evaginations of endoderm. That you can notice there, pharyngeal pouches. So this is how the entire pharyngeal apparatus is formed. You can see there. There are 4 clefts and 4 pouches and 5 arches and 4 membranes. What are membranes? If you just zoom in here, you can see where the inner endoderm and outer ectoderm meet, they are called as membranes. So a pharyngeal apparatus has the 5 arches which are mesodermal, 4 clefts which are ectodermal, 4 pouches these which are endodermal, and 4 membranes where ectoderm and endoderm fuse. That is the first pharyngeal membrane. So these are the salient components of your pharyngeal apparatus. That is how you should understand the development. Remember the 3 to 4 sources there which are formed there, 3 to 4 sources. You can notice like this. So in the textbook you will have a section like this. You have a coronal section like this. You are seeing the, yes, you can see there. If you observe here, this is how you see in the typical textbook diagram. That is the arch. You can see there are 5 arches like that. 1, 2, 3, 4, 6. The 5th one, yes, disappears. And you have 4 external invaginations called as ectodermal clefts and 5 endodermal outpouchings called as pouches, endodermal pouches. And where both endoderm and ectoderm meet, they are called as membranes. You can notice here. So clefts you can see. So these are the cleft structures that you are seeing on the outside. So you can see, and inside you can see the pouches, but the front view they appear like this. You can notice there are horseshoe shaped arches there. That is how you observe. Yes. So this is the pharyngeal apparatus. Just try to recollect the different components of pharyngeal apparatus here again. Hope you are following the animations here. So this is how it is there. And by adding, we have removed the neural tube, but if you add a neural tube here, neural tube, the hindbrain, there will be the ectodermal placodes from the hindbrain, and these neural crest cells will migrate into each arch and they form the nerve of each arch, and there will be arteries from the aortic arch system which are following in the each arch, giving the components of each arch. We'll come to that. What are the different components of each arch? You'll have a cartilage component, a mesodermal component, a arterial component, and a neuronal component, which are giving rise to different parts. So in the schematic diagram, when you observe here, this is the pharyngeal apparatus in coronal section. You can see. So horse-shaped cylindrical bars, lateral and ventral to the primitive pharynx. Okay. So what are the components of arch? As we have seen in the animation, you can see it has a core of mesoderm, a cartilaginous bar, a pharyngeal arch artery, and a nerve which is derived from the hindbrain vesicle that you can observe here. So these are the components of the pharyngeal arch. Yes, this is the section there. You can see the section has gone through the cylindrical embryo in the upper right area, and these are the components of the pharyngeal arch. Each pharyngeal arch has this artery, cartilage, muscle, and these mesoderms. The arch mesoderm will give rise to the muscles of head and neck region. All the muscles of each arch, what we discuss in the future slide, they are derived from this paraxial mesoderm, whereas the skeletal derivatives, they come from the arch cartilages. Arch arteries are derived from the developing aortic arches, and nerve come from the epidermal placodes. Morphologically, each arch will have two nerves: post-traumatic nerve, which runs along the caudal border, cranial border, and pre-traumatic, which runs along the caudal border. And in human beings, all the pre-traumatic nerves disappear except the first arch, where it persists as quad tympani. That's why it supplies the anterior 2/3 of the tongue, It provides a taste sensation of the anterior two-third. It is the only remaining pre-traumatic branch in the human beings. And muscles are largely derived from the mesoderms that we have already discussed. They may remain there or migrate, but they retain the nerve supply of that arch. So if you remember the nerve supply, you can remember the entire pharyngeal arch in a table form like this. The first arch nerves are maxillary and mandibular along with the quad tympani. Muscles you can remember as my must tension anti. My means mylohyoid. Must means masticatory muscles. Tension means tensor tympani and tensor palatii. Anti means anterior belly of digastric. Skeleton are this maxilla, zygoma, temporal bones, and Meckel's cartilage gives rise to these ossicles, malleus and incus, along with the ligaments such as anteligament of malleus and spinomandibular ligament. Second arch, the nerve is facial nerve. Remember, all these nerves are derived from the hindbrain vesicle, that is from the pons and medulla. And the muscles are, you can remember as stupid, stylish facial poses, stapedius, stylohyoid, all the facial muscles, and posterior belly of digastric. Skeleton you can remember as 5S, steps, striated process, stylohyoid ligament, smaller corner of hyoid bone, superior part of the body of the hyoid bone. Third arch, the nerve is glossopharyngeal nerve, and muscle is the only muscle, that is stylopharyngeus. Skeleton is greater corner of the hyoid bone and inferior part of the body of the hyoid, because superior part is derived from the second arch. And the nerve of the fourth arch is superior laryngeal, and sixth arch is recurrent laryngeal. And muscle of the fourth arch are cricothyroid, levator palatii, and constrictor pharynx, whereas sixth arch are intrinsic muscles of larynx, along with the cartilage of larynx. So you can see here, this is how the embryo looks like after the development of the pharyngeal arches. You can remember here the nerves of the arches and the arch cartilage, how they are oriented. You can see here in this picture, you can take a screenshot and remember how these different components, skeletal components of each arch are formed. You can notice they are one below other. First arch, second arch, third arch, fourth and sixth arch. So like this, you can understand the derivatives of the arches. They give rise to nerve, muscle, skeleton of the head and neck. Arch artery, first arch artery is maxillary artery, second is hyoid and stapedial, third arch is common carotid and internal carotid. External carotid is a outpouching of common carotid or outgrowth of the common carotid. Fourth arch artery is aortic arch on the left side, subclavian artery on the right side. Sixth arch will form pulmonary artery on both sides, not only on the right side, it forms on the both side, that is the ventral component, whereas dorsal component forms ductus arterius on the left side, disappears on the right side. And coming to these endodermal pouches, how are these endodermal pouches will form? You can notice here they form different glandular structures and tubes of the head and neck. So the first endodermal pouch will form a tubotympanic recess, which is involved in the formation of the auditory tube, and second pouch will form palatine tonsil and tonsillar cleft. Third pouch will form inferior parathyroid gland along with thymus, which will descend along with the developing heart. Fourth pouch is forming the superior parathyroid gland. You can notice here in this diagram, these endodermal pouches have contribution in the different tubes, lymphoid organs and glands of neck region. So pouches, we are talking about pouches here. We are not talking about the endoderm covering the arch. The endoderm covering the arch have a role in the development of this tongue and pharynx, whereas cleft. So earlier they used to say that the first cleft is giving rise to external acoustic meatus, but now that view is changed because of the research. The first, these all clefts they obliterate. The external acoustic meatus is derived from the first arch ectoderm, not the ectodermal cleft. Remember, these clefts will obliterate and close but if they remain patent, they form an cavity with a opening called as sinus in the neck, that is called as cervical sinus. They may have a duct with them, that is called as fistula. We typically call them as a branchial fistula. You can notice here the cervical sinus may open externally in anywhere with respect to the anterior border of the sternocleidomastoid. Sometimes it may open inside, forming the internal fistula, or sometimes it will not open and form the lateral cervical cyst. So you can notice here, these are the ectodermal clefts and the endodermal pouches. Just remember one thing, earlier ectodermal cleft was thought that forming external acoustic meatus, but it is not the case now. So hope you understood. Thank you.